Genes and Mutations. This presentation is by Charles He, Jessica McClendon, Kaelin Priger, and Wangshu Yang of Group B2.
Are you a mutant? A mutant is an individual that carries a mutation, particularly a new or rare mutation. A mutation is any change in the hereditary material of an organism.
There are several different types of mutations that can occur. A silent mutation occurs when a nucleotide is changed on the gene, but the amino acid sequence still stays the same. A missense or replacement mutation is the opposite of a silent mutation. This occurs when a nucleotide is changed, which does disrupt the amino acid sequence. A nonsense mutation is when the changed nucleotide causes an early stop, and the frame shift mutation adds or deletes a nucleotide.
Genetic variants predicted to severely disrupt protein coding genes, collectively known as loss-of-function variants, are also mutations. They are of considerable scientific and clinical interest. Genome sequencing studies indicate that all humans carry many genetic variants predicted to cause loss of function or protein coding genes. These LOF variants found in healthy individuals may be deleterious, neutral, or beneficial for humans. Distinguishing between these categories will be crucial for functional interpretation of human genome sequences. To study the function of these LOF variants, a group of researchers headed by Daniel G. MacArthur collected data from individuals represented three population groups: Yoruba individuals from Ibadan, Nigeria, Northern and Western European origin from Utah, and Chinese from Beijing and Japanese from Tokyo jointly. They identified candidate LOF variants from these individuals and filtered out those high-confidence LOF variants.
Are you a mutant? Well, from the studies of MacArthur's group, you are. This study looked at 185 sequenced human genomes, and they found out that on average, the human genome has 100 loss-of-function variants, which includes the inactivation of about 20 entire genes. When looking specifically at genomes of certain nationalities, it was noted that the European human genome that was sequenced had 97 loss-of-function variants, and 18 of them were in the homozygous state. As noted before, one of the groups to be sequenced was from Nigeria. These genomes had 25% more high-confidence loss-of-function variants than the other three groups studied that were non-African. This leads to the idea that a greater level of genetic diversity correlates with a higher level of variation in functional gene content. Quintana Mercy looked at MacArthur's findings and relayed them to natural selection through these three hypotheses. The less is more hypothesis looks at loss-of-function variants that are advantageous, and it says that they may increase in frequency because of positive natural selection. The less is nothing hypothesis says that if the loss-of-function variants are tolerated and don't have an impact on survival, they'll drift neutrally through the population. Less is less hypothesis looks at loss-of-function variants that are deleterious, and it says that they'll become less frequent through natural selection because of their impact on survival and fitness.
According to Quintana Mercy's less is less hypothesis, LOF variance between published genomes is complicated by differences in sequencing technology, variant calling algorithms, and gene annotation sets between studies, and by the expectation that LOF variants will be highly enriched for false positives. The basis for this predictive enrichment is that strong native natural selection is expected to act against the majority of variants in activating protein coding genes, thereby reducing the amount of true variation as these sites relative to the genome average, whereas sequencing error is expected to be approximately uniformly distributed. It seems likely that the majority of LOF variants in an individual genome are common variants in non-essential genes, although these may still have an effect on human phenotypic variation. Nonetheless, the signature of strong purifying selection against high-confidence LOF variants as a class, and the discovery of numerous known and predicted severe recessive disease alleles with large effects on human fitness exists at low frequency in the human population. Using LOF-tolerant genes. Some LOF-tolerant genes may have no effect and be inactive. Homozygously inactive genes are an example. LOF-tolerant genes are less conserved and have fewer protein-protein interactions. They also improve chemosensation, which is related to smell.
Loss of function variants can increase the risk of or be the direct cause of several diseases. Such diseases include Crohn's disease, fibrocongenital amaurosis, Tay-Sachs disease, and many, many more. Certain loss of function variants can be advantageous, thereby leading to an increase in population frequency by positive selection. This would support the less is more hypothesis. However, in the case of loss of function variants causing diseases, gene loss would be disadvantageous. This would support the less is less hypothesis. Loss of function variants can also affect things such as the PKD1L3 and PKD2L1 genes. These are responsible for producing proteins that accumulate at the taste pore area of the mouth, which are responsible for the sour taste sensors in us. This can lead to variation in taste sensitivity between humans.
So, you ask, what does this information about genes and mutations have to do with me? Well, now you can go around telling your friends that you're a mutant just like Wolverine or Cyclops. Well, maybe not just like them. MacArthur found that you probably have 100 loss-of-function variants in your protein-coding genes. These variants aren't causing you to be able to regenerate body parts or walk through walls, but they may be affecting your sense of taste or smell, or in a rare case, you may suffer from a disease because of these variants. But you don't have to really explain this to your friends. Just tell them that you're a mutant and they are too. Thanks for watching our presentation. If you'd like to learn more about genes and mutations, please check out the following articles that we've referenced in our presentation.